let's start this session. We have two talks, and after that, I remind you all that we have the posters at 5 at the guest house, so we all have to get there. Okay, first talk of this session will be given by Simone Pigolotti, and that will be about variations of speeds of bacteria. So, please.
Thanks a lot. Can you hear me? Good. So I'll be happy to take questions during the talk, as usual. To my great surprise, mine is not the only talk in this conference about DNA replication, after the very nice talk by Anjana yesterday. But I came about thinking about this problem together with Pablo, also in the audience, about 10 years ago, maybe, from a very different angle. So it's a bit more in the flavor of statistical physics. So if you think about DNA replication, fundamentally, you have a heteropolymer that encodes some information, and there is an enzyme that tries to replicate it. And there are two reasons why this is interesting. One is that this is perhaps the most fundamental task that cells have to perform. If they cannot do this, they cannot replicate, and they cannot transfer genetic information. But also, there is another reason, which is related to thermodynamics. So if you think about the quality of this process, ideally, you would like to perform this task with a very high accuracy, so produce a reliable copy of your template without wasting too much energy. So ideally, you would like your dissipation to be low and at very high speed, because the faster you do it, the faster you can replicate. If you were doing this at thermodynamic equilibrium or close to thermodynamic equilibrium, you can easily estimate that the accuracy will be low because the energy differences of right matches and wrong matches between monomers is not so large. There will be no dissipation because we don't dissipate at equilibrium, and speed will be low as well. So if you think about this in this way, you can think that these machines, what they're actually doing is trading thermodynamic energy for accuracy and speed. And there are lots of interesting questions on the optimal way of doing this. So if you, for example, you want to spend more energy to increase your speed or to reduce your error, and whether there is a trade-off between speed and error, and so on and so forth. So what we have been working on is to use ideas from stochastic thermodynamics and non-equilibrium thermodynamics to understand optimal trade-offs between these quantities and optimal designs of the biochemical networks that perform these operations. And it turns out that you can develop a rather elegant theory, but unfortunately, the physical limits of the operation of the systems tend to be not very tight bounds to what real biological systems do. And that begs the question, can we use the body of theory that we developed to, and in general, ideas from non-equilibrium statistical physics to say something about how these machines actually work. So I will show you in my talk an example of this. So this is a bit of a different perspective from this work, in which we are a bit more based on what the experiments tell us. So the system that I will talk about is DNA replication in bacteria. And for those of you, I apologize with the experts, but for those of you that are not expert in this, I will give you the basic facts. So this is the most common architecture. Of course, in biology, there are always exceptions, but this is what's typical for bacteria. So often bacteria have their genome organized in a single circular chromosomes. And there is, wrong button. So there is a replication starts at a well-defined site called the origin. There are two machines called replisomes. There are this big molecular assembly that replicate the genome. There are two of them. They both bind at the origin, but they proceed in opposite directions. So one it goes clockwise, and the other goes counterclockwise. So each one of them replicates both strands of DNA. So in this figure, all the lines you see are double-strand DNA. The red will be the template, and the blue will be the copy. But this distinction is somewhat arbitrary because each one of them has one strand from the template and one strand is new. And so these two machines will meet at some point opposite the origin, thereby finishing replication. So this is essentially how things work in most cases. And our goal is to say something quantitative about these dynamics. And in particular, our initial curiosity is, can we say something about the uncertainty on the meeting point? So this goes in the direction of thinking about accuracy in biology, for example. All clear so far? Questions? Good. Yes?
So in principle, they can meet at any point, or they always meet at the terminus of replication, so at the opposite side compared to origin?
There are some details that I just didn't mention. So there are some sites that limit the region in which they can meet. So essentially, there is a limited region where the meeting point can occur, but there's not a specific site. So there is an uncertainty within a certain region. That's the short answer. Yes?
Another question. So, as a physicist, I understand very appealing this idea that there is an optimum that maximizes accuracy, speed versus energy. However, I find that sometimes in biological community, researchers like to challenge these principles. For instance, if a bacterium is so rich in food, could it be that some species under some conditions actually do not care about how much energy they spend, but they prefer to explore the maximum limits of speed?
Of course. Of course, that goes exactly in the direction of which I'm saying, which is maybe we should. I'm not talking about first principles in this talk, but more about let's see what we can learn from actual experiments. So, so this is the background. So the idea of our work has been inspired by experiments that I've been doing in the last maybe 10 years, a bit more. And this is a very simple experiment. So here you grow a bacterial population in the exponential phase. You take a sample and you sequence it. And you look at the abundance of DNA fragments along the genome. So because replication always starts from the same point, what you see here is that the abundance of fragments is not uniform, but it has a peak around the origin, and then it falls down. So this makes sense because if you have an exponentially growing population, there will be a lot of incomplete genomes. These incomplete genomes will always contain the origin, and they will have a decreasing probability of containing positions that are farther and farther from the origin. So we want to essentially expand on this idea, and yes?
Does this matter? Does this depend on how many copies of the genome each bacterium has got, or?
I'll get into that in 1 minute. Thanks. Okay, so that's the basic idea. But there is a small complication, and this has to do with the relation between DNA replication and the cell cycle. So in slow growth condition, and again, this is a bit of a review of basic biology of bacteria, but again, if you know it, I apologize. If you don't know it, this is important. So if you are in slow growth condition, the cell cycle of bacteria is characterized by 3 different stages that are actually organized depending on DNA replication. So there is a stage B, which is a pre-replication stage, where the bacterium contains only its template, its original genome. Stage C, where replication occurs. So we have a template plus an incomplete genome. Stage D is the post-replication phase, where inside a bacterium you will find 2 complete genomes. But if you grow, for example, E. coli in very fast condition, it turns out that the cell cycle can be shorter than the time it takes to replicate the DNA, which means that DNA replication has to be initiated in the previous generation to be passed to the next cell. And so what happens here is that at birth, the bacterium inherits a genome that is already undergoing replication, and then it will start replication again during its lifetime, and then it will be passed already undergoing replication. And you can imagine that this can get quite complicated, and it's not so comfortable to model. And so what we did is to say, okay, if you have a growing population, you will find cells at different stages in their cell cycle. They will contain a different number. So I always use this color coding. So red is the template, blue is the replicating genomes, and green is the post-replication genome. And again, there will be all these possible complicated configurations that there can be. And so what we want to do is ignore cells. So just look at the genomes, and let's think about what happens at an ensemble level. So if I have many cells that grow asynchronously, then essentially you can formulate a model like this, in which you have these three types, these three types of genomes, the incomplete ones, the templates, and the post-replication ones. And you will produce genomes with certain rates. So K is the rate at which you initiate replication, so you create a new synthesizing genome. Beta is the rate at which you complete replication, so a synthesizing genome will become a post-replication genome. And then cell division will transform a post-replication genome into a template, okay? So this is just a simple population model, but the idea here is that we are doing it at the level of genomes. We are not looking at what cells do. But incidentally, the model takes into account the number of cells, because you can see here that no matter what happens, each cell always has one single template. So the number of templates also is equal to the number of cells. Okay, so now the tricky part of the model is that now we want to zoom in this synthesizing genome and say something about their size spectrum. So what we do is to say that, so if I call x1 and x2 the length copied by each one of the two replisomes, I'm assuming that replisomes travel, the dynamics of replisomes is described by a certain Langevin equation that for simplicity I assume to be the same for the two replisomes, and it will contain a deterministic part with a velocity that can in principle depend on the position, and a fluctuating part, which is essentially one-dimensional diffusion. And so if you do the math about this model, so there is a coupling between these Langevin dynamics and the population level dynamics of genomes. What happens in the end is the following. So if I look at the population level probability of finding an incomplete genomes with a length x1 of the genome copied by the first replisome and x2 by the second replisome, this will evolve according to a PD, which looks a bit like a Fokker-Planck equation, but has a death term, which comes from dilution, so from the fact that population is growing exponentially, so you don't have a conserved number of cells. So this equation can be, in simple cases, depending on the choice of the velocity and the diffusion coefficient, can be solved analytically, or it can be solved numerically using an analogy with the stochastic resetting. So I can interpret the dilution term as a resetting term where I follow these dynamics, and then I can restart essentially from the origin. The phase space of this problem is this triangle, because the process is completed when x1 plus x2 is equal to the genome length. And so essentially, you have this Langevin dynamics with a resetting term and with an absorbing state at DNA completion. So this is essentially the space in which this problem lives. So clear so far? Okay, so essentially what we do is look at the steady state of these dynamics, and essentially, oops, sorry, I always have a problem with this. So this steady state will tell us the size spectrum of incomplete genomes, and from the flux through the absorbing boundary, we can also infer the fraction of complete and incomplete genomes. And so it's quite easy once we have the steady state to predict the distribution, size distribution of DNA fragments. So this is essentially a pictorial representation of this equation. So if you think about the probability of finding the number of genome fragments of or at a certain genome position, you can see that the templates and the post-replication genomes will give a constant uniform contribution to this distribution, whereas the incomplete genomes are responsible of the size spectrum, as you would expect. So as I said, this is nice because this is a pattern that we can observe in experiments. And also, the nice thing is that sequencing techniques are improving quite rapidly. So we actually repeated these experiments at OIST, and we could get much more precise curves. So this is E. coli growing at 5 different temperatures, from 17 degrees to 37 degrees. And you can see that with some tweaking nowadays, you can get very precise data. So this is great because now we are in the position to do inference. So if I choose my Langevin equation, my theory gives us this curve. I can measure it into experiments, and so I can go back and find the best Langevin equation that describes the dynamics of replisomes in the best way. Clear so far? Good. Okay. So clearly, the simplest assumption we can make is that replisomes travel at constant velocity without diffusion. So in this case, the model can be solved analytically, and we get that this distribution is simply an exponential. This is the fit. I show you just 37 degrees, but other temperatures fit similarly. You can see the fit is pretty good. The distribution depends on 2 parameters. One is k, which is the exponential growth rate of the population. The other is v bar, which is the velocity. We can measure in the experiments the exponential growth rate directly by just looking at the optical density. And therefore, with this fit, we can get the replisome velocity v. And we find that in the range of temperature we explored, this dependence is well described by an Arrhenius law. This is pretty similar to what happens for the growth rate itself, except the effective activation barrier is slightly different. This is really the Arrhenius plot, so I'm plotting both the velocity and the growth rate versus 1 over t. You can see in logarithmic scale, so you can see that these two have 2 different slopes, and there is a deviation at 17 degrees in both cases. So this makes sense because this essentially is telling you that if you increase the temperature, the growth rate grows faster than the speed of replisomes, which means that essentially replisomes cannot catch up with the speed up of the growth rate. And this is a good justification of the fact that this multi-round replication system is necessary. So if the velocity could catch up with the speed up of the cell division time, then everything would be synchronized, and there wouldn't be a need for this system. So the idea here, I mean, we are among friends, so yes?
Hi. At some point, you showed population dynamics model with the alpha and beta transition rates. Where do they enter in the predictions here?
That's a very good question. So in the end, we do not really need to solve this model. So we have this model. Essentially, we really solve this dynamics, not this dynamics. But we need to couple this thing because essentially we need to connect the rate of completion with the fraction of complete genomes. So essentially the need for this model is that the rate of completion beta enters this formula here. So we needed to say that essentially the fraction of complete genome is beta divided k over beta. And we can measure beta from the stochastic simulation. So essentially, yeah. Thanks. Suri?
Like plots for growth.
Yeah.
Have they always been in the exponential phase, or do they are those Arrhenius-like plots for growth that others have published? Are they in other cell phases?
So the one for growth is well known. We found the value of the activation barrier slightly different. I don't know. I think usually people report 24 kb. This is 22, something like this. But it might be the broth. So, the growth medium is slightly different, so, there might be a small dependence there. Thanks.
Simone,
Yes.
maybe related to this. Is there no ATP consumption in the movement of the replosome?
Yeah, of course. Yeah,
Then
this
how
is
is
really,
it that Arrhenius is so good at room temperature? For V bar.
I don't know. I mean, lots of things here, lots of complex reactions in biology are described by Arrhenius law, even if they are not simple reactions. Why is that? Somehow there is an effective barrier, but I don't know exactly. There could be one rate-limiting reaction, of course. Yeah. Okay. Yeah, so what I was saying is, again, just being honest here. So our idea was, well, if we solve the model with diffusion, of course, we have an approximate solution, but you can sort of solve the equation in your head at least to some extent. You can imagine that here, this solution has a cusp here and a cusp here, and the effect of diffusion would be to smoothen the last part of this distribution. So if we could see how smooth this is, then we get a sense of the diffusion, and that's nice because then we know our uncertainty. But then we started looking at these curves, and we looked at that actually there was something else that caught our attention, and it's the following thing. So if you see here, you can see that here the points are a little bit above the curve. Here they are a little bit below, and then they're a little bit above. You might think I'm going crazy here. But let me magnify this effect by plotting instead the data divided the theory, divided the exponential solution. So if I do that, this is what we get. This is the 5 different temperatures that we studied. 17 degrees, this looks pretty much like noise, which means essentially the model is fitting well our data. But at increasing temperature, you start to see. So this is the whole genome. The whole genome is 5 million base pairs.
It's 1 megabase between each.
Roughly, yes. So you can see that there is a regular wave-like pattern. It's symmetric, so in these plots, the origin of replication is in the middle. So this looks like something that is affecting the 2 replisomes in the same way. And it's highly repeatable. So we repeated these experiments at the same temperature 3 times. You always see this. So this is clearly not fluctuations, right? This is a systematic effect. We don't have it at 70 degrees, but it's present at all other temperatures. So we said, yes? That's a good question. We didn't try different media. We stuck with LB because we have a nice exponential regime. But yeah, we should try different media. That's a good point. So we said, well, this is what we see. Maybe let's try to describe it. We have a flexible theory. So the idea is let's have an oscillating velocity model.
Yes. Can you go back to the previous slide, please?
Of course.
Can this be the kind of graph, it looks like GC skew graphs. GC skew, the amount of skews in the genome. Has it been correlated with the amount of
we
Gs?
tried. It doesn't look the same. The length scale of GC skew is much shorter. Okay. We don't have such long wavelength oscillations in GC skew.
Okay.
Thanks. Can I continue? Oh, yes.
So, related to that, can you do that in different organisms that they have more or less GC content to see whether?
I would like to. I didn't do it yet. That's a good question. Can I'll continue. Okay, so, oh yeah.
I mean, there's no symmetry of the genome, is there? On either
No,
side
not
of
at all.
the...
Not at all. But I'll go into possible explanation in a few minutes. So first, we say, well, let's model it. I mean, we see it. I mean, let's do a phenomenological model. So again, our framework is flexible enough to describe this. So now we say our velocity is not constant along the genome, but is a constant plus an oscillating term. So delta will be the amplitude of these oscillations. And then this thing will have like a wavelength and a phase. And in this case, we say, since we are doing a more general model, let's throw in diffusion as well. And so in this case, we couldn't solve the Fokker-Planck equation with resetting analytically, so we just did this numerical simulations. And it turns out that this model fits very well the data. So now I can show you. These are the solutions for the DNA abundance. So you can see that it's not really exponential. It's like a modulated exponential. And this is with this discrepancy ratio. So you can see that this model fits this wave-light pattern very well. We have to be careful because now we introduce the extra parameters, but the information criterion always says that the improvement in the fit is worth the extra parameters of the model. So statistically, this is solid. So the outcome is, let me first discuss the oscillations, and then I'll talk about the diffusion. So what the model is telling us is that the velocity of replisomes has this wave-like oscillation along the genome with this very long wavelength, symmetric, so both replisomes experience the same oscillations, and the amplitude is something on the order of 15%, yes?
Could you explain the connection with stochastic resetting that was on the...
Yeah, so this is the equation we get from the model, and K comes from the fact that you have a exponentially growing population. But really, this is the same equation you will get for drift diffusion with stochastic resetting. So instead of having a growing system, like starting new system over and over, we have a single simulation in which we reset to the original rate K. Yeah, a stationary state, yeah. Thanks. Okay. Yeah. So I was telling, this is what the model is telling us. And again, in full honesty, we were like, can we publish this? Like, this is what the model tells us, but this is a bit crazy. Why should there be these oscillations? Again, as Joe was currently saying, the two replicors are completely different. So why also you would expect these similar things in the two sides of the genomes? And so something that really comforted us is that we discovered that people have been, in recent years, studying fluctuations of the mutation rate along the E. coli genome in mutagenesis experiments. Can I finish this? And what they found is this. So also the mutation rate along the E. coli genome displays wave-like variations. And these variations are highly correlated with the pattern we find. In fact, the correlation coefficient is something around 0.75. This is quite remarkable because these are experiments done in different conditions, in different labs. It's not even the same strain. So this is, again, comes back to the origin. This is highly suggestive of a speed-error trade-off. So the idea that in regions where replisomes travel at a faster velocity, they will also commit more mistakes, and so as a consequence, the observed mutation rate is higher. Yes, please, sorry.
Is this correlating with the, the cell division rate or the cell cycle time? I mean, how often it's dividing? Because if it divides, it'll affect the replisomes going in both directions.
Not precise. I will show you what's okay. So yeah, maybe this is what I guess I wanted to say. So of course, the question is now what are the causes of this? And our idea at the moment is that these oscillations might be caused by this multi-round replication. So you can imagine that if you are a replisome, you're traveling along the genome, and if you are in fast growth condition, at some point before you finish, another replisome will start replication. If there is some degree of competition among replisomes, for example, for nucleotides or for other enzymes, then it might well be that when the next round of replication starts, your velocity is decreased because you're lacking some common shared resources. Yes?
You might come to this might be a crazy idea. So in the beginning, you talked about speed error trade-offs in the first thing. One aspect that that class of models typically ignores is the geometry of the DNA itself. So if you make an error, I'm imagining that there is a region with higher stress locally, and I'm imagining that the replosome can't travel as freely if you have higher stress-like conditions. Can that kind of a thing sort of give you oscillations in velocity? Wild thought, sorry.
I don't know. That's a good point. I'm not sure I would test that. Maybe we can discuss later. Okay, so essentially what I'm saying is that the model is, for example, telling us that 17 degrees will be in a regime in which this multi-round replication does not happen. So on average, you'll have less than 2 replisomes per genome, whereas in at all other temperatures, you would have multi-round replication in your experiments. And so the fact that we observe these oscillations for these 4 temperatures, but not for the lowest one, is an indication that possibly multi-round replication is the cause for this. And also the wavelength we find is consistent with the expected time between replication starting events. But again, these are just suggesting evidences. They're not, we don't have a final proof, so more work is necessary to understand how this should go. Okay, so the last thing I want to show you is the results for the diffusion constant. The diffusion constant happens to be small. In fact, in a few of our experiments, essentially this is compatible with 0 in the sense that the information criterion tells us that we are not justified in adding the diffusion constant as a parameter. But for many other cases, we get finite fits, in fact, for the majority of cases. And we get values of on the order of 1 kilobase per square per second, which is quite small. In fact, is comparable with equilibrium estimate for the diffusion constant for a macromolecule of the size of the replosome. So this, if you put it on a back of the envelope estimate for the uncertainty of the meeting point, gives you something on the order of 100,000 base pairs, which is consistent with other experimental evidences. So this seems like a reasonable number. Okay, so I reached my conclusion. So we introduced this method to study the dynamics of bacterial replisomes in vivo using these simple ensemble experiments. Our theory, our method tells us that the average replosome speed depends on temperature in an Arrhenius way. And we found these surprising speed oscillations along the genome that are strongly correlated with the mutation rate. And the diffusion coefficient of this machine seems to be quite small. There are lots of things we want to do in the future. One is use this observation to say something about the speed error trade-offs for replisomes. There are other things we want to explore about the biology of replisomes. For example, something that is quite interesting is why bacteria did not discover replication using multiple origins. So they have these long genomes, some of them, and it would be quite advantageous to start replication in different ways so that essentially you could finish the job in a quicker way. And to understand this, some labs have engineered strains of E. coli having multiple origins of replication. These strains are viable, but they have a lower fitness than wild-type E. coli. And these are preliminary experimental results in our lab. So we want to use our theory to describe these curves. Okay, I conclude here. I would like to thank my unit at OIST, Deepak Bhatt, that now is a PI at Vellore, and now Florian Pflug are the main actor in this. We have postdoc positions available, both theory and experiments, to work on this problem. My external collaborators for the first part of this project, and my funding sources. And thanks for your attention.
Thank you very much. It's time for questions, and as always, we will start with questions from students and postdocs.
Hi, Simone. Thank you for the nice talk. So my question is regarding the stochastic resetting part. We know that when we have this stochastic resetting, so if I think of the first passage problem, so there is some optimality, right? So depending on the resetting rate. Now, if I see your equation, your K seems to be related to the resetting rate.
It is the resetting rate.
Yes. So now my question is that, so seems if I think of one replication process, the space seems bounded. So depending on the resetting rate, what is the time scale of a replication process? Now, if K is increased, is it possible that I will not replicate?
Well, I mean, it's a good question. I am not sure. So first of all, you have to think that this resetting thing is only an analogy. So this is not a resetting problem. Really what's happening here is that the number of cells or genomes is growing exponentially. And somehow this is a statistically equivalent at steady state to a resetting problem. Whether the optimality, also the other thing is that this is a heavily biased problem. So the diffusion constant is small, the drift is large. So this is a bit of a different regime from standard stochastic resetting. So whether the optimality principle has, what's the meaning in this context? I'm not sure. I would have to think about that. But okay. It's a good observation.
Okay, thank you.
Thanks.
Simone, on this question of whether or not the diffusion coefficient is large, you said you compared it with the free diffusion of a colloid of similar size. On the other hand, there is this Arrhenius-like behavior. So wouldn't you have to compare with diffusion in a kind of corrugated potential or so?
Well, the thing is that I don't know what the potential is.
Yeah, but I mean, if there is some potential landscape, I mean, you talk about this activation energy. So I'm just wondering whether the statement that the diffusion is unusually small should be so surprising if you compare it to free diffusion.
You're saying yeah, that's a good point. So if I interpret that as really a barrier, yeah, that would be a nice estimate to do. I mean, I was thinking like the bulk diffusion, but yeah, also that could be a nice, thanks.
Just to follow up, Udo, what about space-dependent diffusion? Because you have like absorbing site, no? So doesn't it make any sense to you to have a space-dependent diffusion?
Possibly. The thing is that since the diffusion constant is small, we'll never be able to reliably fit. So you have already a small constant. If you have a variation, small variation of a small constant, we will never have the resolution to fit them. So, yeah, thanks.
Yes. Don't you think that it's a sequence dependent? So there are some locations where error is not so important, you can move fast, but some locations you have to spend time more. If you look at your graphs, the peaks of the errors in similar locations, am I right or wrong?
Yeah. So I would more argue the other way around, that I think something that would be also interesting to see is whether somehow essential genes are placed outside the peaks. But I can't imagine how the sequence could drive this, because on this length scale, everything is average. So, you know, we are talking about hundreds of thousands, if not millions of base pairs. So we haven't found any signature in the genome that could.
A very nice talk. I was a little bit surprised by. Yeah, I was a little bit surprised by your explanation for this in the context of replication initiation, right? So E. coli, in this overlapping fork regime, it fires these origins synchronously in concert. And when that happens, with respect to the cell cycle time, the phase at which this happens, that depends on the growth rate, but that varies continuously, right? And so you wouldn't, so I think this explanation that it has to do with when you start new rounds of replication, because I thought that was your explanation, no? That it could have to do with when you start a new round of replication, that you then have competition, but I don't see how you could then get these oscillations, right? Because I think when it starts and where it starts, that depends on the growth rate, and that varies continuously. So I don't see how you could get...
But it's continuously with what?
With the condition, right? So, right? I mean, so under a given condition, right, a new round of replication starts always at the same point in the cell cycle.
Yeah.
And that starts then, that also corresponds to a specific configuration of your DNA, if you wish.
Okay.
So I don't see how you could then get this oscillatory pattern out of that.
But no, I think that's precisely my point. So if you have a timing, like a precise timing between different rounds of replication, if you multiply this time by the velocity of replisome, that gives you a length scale, which is not the entire genome.
Yeah, but
so
I don't
just
think
saying
that's
that
a characteristic. Well, so there is a characteristic, right? I mean, the time between new rounds of replication, that is always the cell cycle division time, right? That's always the cell cycle time, right? So per cell cycle, it starts a new round just once, but then it fires all of these origins synchronously.
Yeah.
Time in between
yeah.
firings is just the cell cycle time. And at that moment, when a new round starts, you have a specific configuration of the DNA. So how? Yeah. So I don't quite see how.
So this time, multiplied by the velocity, will give you a length scale.
Yeah. Or is it okay? So it has to. So it has to do with the differences between the length in the DNA between the different origins at a given round of replication.
Yeah. Yes.
Initiation. So you start a new round of replication initiation, then you look at the configuration of the DNA. It has this complicated structure with these overlapping forks, and then you measure the distance between the length of the DNA between the different origins. That's how you get this out, I think.
Somehow, yes. Okay. I guess. I don't know if you're understanding each other, but yeah.
But I have one other question, a very quick one, much easier one. So that's on the diffusion, right? So you have these two strands, right? And in one strand, that's the easy one, and the other one is always sort of, copying backwards all the time, right? And so you would think that the diffusion constant between these two is different, right? So if you replicate from the origin...
So we are at the larger scale, right? So the replisome copies both. Each replisome copies both strands. So copies the leading strand. It has one leading strand and one lagging strand, which is the opposite for the other one. But each one is copying both. So that happens inside. Yeah.
Sorry, another quick question. You were speculating that the competition might be on nucleotides, but those are the molecules that are in the highest copy number. Wouldn't it be more plausible that it might be something in the replication fork machinery or something?
Absolutely, it can be spare parts. So these machines are dynamic. So sometimes a piece falls off and needs to be replaced.
Right.
So doesn't need to be.
Those are much lower copy numbers than the nucleotides, right?
But they're also substituted more rarely. So I don't know what's the. It could be either. So I think this. I wouldn't bet on either of them.
The last two questions, which are already reserved.
Thanks.
So what is the relationship between the cell cycle and the replication cycle? Are they coupled, and, you know, cell division and replication?
So they are, it's complicated. So it has to be once, but the time is different, so they are somehow staggered, right?
Yeah.
So I don't know if that answers. Yeah, maybe we discuss.
So basic question about how DNA replication works. So doesn't the speed of replication depend on the concentration of nucleotides in the surrounding medium?
Yes.
Okay, so maybe...
Yeah, phosphorylated nucleotides, yes.
Okay, and you said that in bacteria, the replication happens, so the chromosome is a ring, right? But the chromosome is a ring, but it's not necessarily like a ring in 3D, like it might be coiled many times around itself, right?
Yes.
So if there was a non-uniform gradient of nucleotides in the cell, then wouldn't it accelerate when it passes by a high concentration, then decelerate when it goes back on the other way, or when it coils around many times? Okay, okay, but I was thinking maybe it's not maintained. Okay, okay. Okay, all right.
Okay, I think we we have to move on because of time. So let's thank Simone again.